We have 6 exciting lectures, and each lecture will be exactly 20 minutes, so we really have to start on time. And I would like to invite the first speaker, which is actually me. So I'll start. Okay, so my talk will be about UV disinfection of coronavirus, surface water and air, and actually I will mostly emphasize air and surfaces and not water this time. The Ashley organization actually stated that airborne transmission of SARS-CoV-2 is significant and should be controlled. As an engineer, there are many ways to control viruses, which could be related to the operation of heating and ventilation in air-conditioned systems. Now, the strategies actually to mitigate, is this working? Okay. The strategies to disinfect, to mitigate air contamination could be by bipolar ionization, filters, electrostatic precipitators, photocatalytic oxidation, vaporized ozonation, vaporized hydrogen peroxide, and UV. Basically, as being part of the committee of the Ministry of Health for technologies to actually mitigate contamination in closed rooms, one of the requests is that we'll have zero ozone emissions from air cleaners. So this is a critical request, and all the technologies that generate free radicals or reactive species have the potential to actually emit ozone, and therefore this should be analyzed. We will target in this discussion UV-based technologies, which could be, for example, controlled by filtration, followed by UV disinfection. Now, UV is actually a broad range term for different types of lamps that emit UV. So, for example, we have mercury lamps that can emit UV at 254 nanometers, and this is the simple lamps that are actually in our home appliances. We also have polychromatic lamps, and we also have the new technology which are termed LEDs, and we can see here the LEDs that also can emit in the UV range. I will emphasize in this talk air disinfection and surface disinfection. So just let's start about talking about air. Well, the ASHRAE also advocated using UV to target disinfection, and we can see that there are many places where we can actually apply UV. So, for example, we can apply UV if we look here at A. We can apply UV in the air ducts. We can apply UV in upper air disinfection. We can apply UV, for example, in various surfaces. And the UV will actually inactivate the microorganisms. And these are actually images of the different types of places where UV can be actually implemented. So this is in-duct disinfection. This is, for example, disinfection of the coil and not the air itself. And these are, for example, portable room decontamination. And actually, since all the corona crisis started, there was a boom in the different UV technologies that could be used, even handheld UV, which is actually very dangerous. And this is pictures taken from Tel Aviv University. And in one of the halls, the student complained about smell, probably from bacteria, mold, and fungi. And therefore, the university actually implemented a UV technology here, but this is for disinfection of the coil and not the air itself. And these are, for example, ventilation systems at our university. And these are actually could be designed to add a disinfection unit from the air that actually circulates through the building. Now, the guidelines for adding UV in ventilation systems or in ducts could be actually looked upon the EPA, which is the Environmental Protection Agency, or even the ISO standards. So there are ways to actually evaluate the UV dose in these systems. We need to target a certain minimum dose. So this is one of the key parameters. we need to have a minimum airflow, and we need to have a minimum irradiance over a certain area, and we need to have also minimum exposure time of 0.25 seconds. And it always should be applied or coupled, so UV should always be coupled with mechanical filtration. And the different model viruses that could actually be used instead of the SARS-CoV-2 virus is, for example, the MS2 virus, which is also single-stranded RNA virus that is non-pathogenic, and it's a bacteriophage. So what we did, we combined together a project with Electra, and this is called the Electra-Tau partnership to establish corona laboratories. We submitted a proposal on actually designing disinfection systems through air ducts. This is a project that is done together with Professor Alex Lieberson. And basically, we designed an air duct. And this air duct, actually, we have here an air channel with a size of 60 by 60 centimeters over 1 meter length. We have here actually a suction filter. We have a blower. We have an aerosol generator. So the aerosol generator will actually generate aerosols of viruses. Here we have the UV system. And here we have the air sampler. And the flow velocity would be 4.5 meter per second, which is actually 6,000 cubic meter per hour of air. And this lab is actually near the medical school. So now the next step was actually to design how would we situate the lamps around the air duct. So this is, for example, our first design where we're using 8 lamps. Each lamp is 95 watts in order to generate enough UV to inactivate the viruses. However, the UV could be implemented in various ways, either along the parameter or perpendicular to the airflow. We also tested different materials for the design. So for example, here we used a material that has a high reflectance, and the idea to have a high reflectance is to reflect the UV photons around the lamp. So our model virus was MS2. We did various preliminary testing. For example, we wanted to know that we can actually recover MS2 from the air sampler. And we wanted to understand that we're generating enough aerosols. So we actually prepared a solution of a fluid solution with MS2 viruses. And just please note that small aerosols are aerosols that are smaller than 10 micron. Above 10 micron, we actually generate droplets. Now, the problem with generated droplets is they may actually settle down in the air duct instead of being aerosolized in the air. So this may be a problem. So we generated here, I don't know if you can see it very good in this picture, we generated the aerosols, we inactivated them, and then we actually measured, and we saw that we have a good recovery of the aerosols. So this is a CFD model of the lamps. Here we have 2 lamps, 2 lamps, 2 lamps, and 2 lamps. And this is here the model. We saw that we have like, this is the average dose that we got, and we actually wanted to have 2 logs in activation, which is 99% removal of the MS2 virus from the air duct. Actually, we got 95%. So our next step is actually to improve the process by adding either more lamps or changing the lamp configuration. So this is a working process that is done together with Electra, Tau. So this was one project and about disinfection of air. If this procedure will work, we'll actually generate a small air ducts that could be actually inserted into air circulation units, and the collaboration with Electra is actually really interesting because we learned a lot with them about how to design these systems. Another way actually to disinfect is by using UV LEDs. So LEDs, we know LEDs from the visible light range. We know that we have LEDs in all the different colors, blue, green, red, and so on. LEDs are different than mercury lamps because they're just like the switch of our light. They turn on and off, and so they have they reach their maximum power immediately. Okay, LEDs currently have lower performance and higher cost, But if we look at the market trend, LEDs will, their cost will be actually reduced in 1 order of magnitude over the next 2 years, and therefore probably UVC LEDs will be used as a disinfection in the future. UV LEDs could be integrated in different designs. So for example, we can integrate light into keyboards to disinfect them while they're not being used in hospitals and so on. In this study, we actually tried to examine the impact of different wavelengths on inactivation of MS2 bacteriophage. And what we saw is that, just to go really quickly, what we saw is actually that 265 nanometer was the wavelength that was mostly efficient in disinfection of MS2 from glass surfaces. Then we tried to, and this work was done together with Michal Mendelbaum and Nechama Friedman from the Center of Virology Laboratory at the Sheba Medical Center. And we tried to examine the impact of the different wavelengths for inactivation of human coronavirus. Now, with MS2 virus, we saw that 265 nanometer was the most efficient wavelength, but now we wanted to see what happens with the human coronavirus. And so to test this, actually, we wanted to use a surrogate for SARS-CoV-2, and the surrogate was this virus, and actually, which is less pathogenic than the SARS-CoV-2. However, it has it has structural similarities, and therefore we use that as a reasonable surrogate. And we tested the inactivation for different wavelengths. And again, what we found is that 265 nanometer was the most effective wavelength out of the different wavelengths. However, everything has a cost, right? So we examined the different wavelengths, but the 285 nanometer LEDs are much lower in cost compared to the 265. So although this is much more effective, we can actually think about using this LED and increase the exposure time. The next study, which actually the results were summarized only yesterday, was to test the different variants. So in this study, we tested wild type, the Delta, the Omicron, and the Alpha. And again, and here we tested only one wavelength, the 265 nanometer. And all of the results, we're still working on the data and the results. It seems first that the wild type is actually more resistant. The wild type is actually more resistant the wild type here is actually more resistant to UV compared to the Omicron, the Delta, and the Alpha. However, if we look at the exposure time is about like 10 to 12 seconds in order to inactivate the coronavirus. So actually, it's a much more sensitive virus compared to MS2 bacteriophage. So these results were actually very surprising. So a really low dose of 2-3 millijoules per centimeter square would be sufficient to reduce 3 logs, which is 99.9, just look at the time, which is 99.9% reduction. And then the next step actually was to see if we can design, actually transparent high-touch surfaces that can actually self-disinfect. So this work is in collaboration with Professor Kimberly Gray from Northwestern University. In this case, we actually activated a photocatalyst using UVA light. So the activated photocatalyst actually generates reactive oxygen species that can also oxidize the proteins and the lipids and the DNA inside the cell. So for this, for achieving this goal, we designed a system that had actually 100 LEDs. These LEDs are actually at 365 nanometers, so they're in the UVA range. And we wanted to test the efficiency of disinfection using this technology. So, and just remember, we wanted to design a cover that would be activated by UVA visible light and that would be transparent. So for example, it could be used even like in our cellular phone that will be self-disinfecting. So we used different types of methods to coat the surfaces. And then what we saw is that with most of the catalysts that we used activated by UVA light, we got at about 70 minutes of exposure, we got 2 logs in activation, which is 99%, and we needed like an hour and a half to have 3 logs, 99.9% in activation. So most of the actually surfaces worked really well, but again, we wanted to use the airbrush technology because we're very interested in having a transparent cover and surface. And again, one of the things to note is that when we did, for example, our study, also with the surfaces and also with the air disinfection, we used a buffer. However, we know that these viruses are actually contained within saliva, okay? So the liquid itself can absorb part of the UV photons. And if we see here the absorbance pattern of saliva, we can see that has a maximum at about 290 nanometers, but it does absorb a lot at 280 nanometers. So now our next goal is to see what would be the inactivation efficiency of using all these technologies. So using also the filtration and the low pressure UV within the air duct, using direct UVC for surfaces, and using photocatalysis that is actually activated by UVA light in the background of saliva. And yeah, I'm on time. And thank you very much. And that's my lecture. If someone has a question, because I have 2 minutes. If not, then we'll go to the next speaker.